Hello students, welcome back to the biology class. So in the last class we did the topic of replication of the DNA. We understood that Watson and Crick were the two scientists who postulated that DNA replication should be semi-conservative, but they only theoretically stated that. What made them make this statement was because they knew about the complementarity rule, and we learned in the last class that it is very easy if the one strand information is known to us. The other strand information can easily be depicted because there is a rule of complementarity that C always will come opposite to G and T always will come opposite to A, and the vice versa. We learned that when two old parental strands undergo separation, they will cause the formation of the newly synthesized strands, and those strands are going to be made by the DNA polymerase enzyme. We learned that when two strands separate from each other and when two new strands are formed, then the resultant DNA molecule will carry half, or you can say one strand will be the old one and the new one will be one. And in the same manner, the other molecule of DNA will carry one old strand or the parental strand, and the other strand would be the newly synthesized one. That means it would be called as semi-conservative mode of replication because half of the DNA is old and half is newly synthesized. This was experimentally proved by Messelson and Stahel when they performed the experiment in 1958. We learned the experiment they performed, which had the steps. First of all, growing the E. coli for many generations in a medium of nitrogen salt, which was the heavy isotope of N15, ammonium chloride. And once the DNA has undergone replication, after many generations, what will happen is every bacterial genetic material would carry only N15, so heavier isotope. Now one of the bacterium was taken out, its DNA was centrifuged in cesium chloride containing medium, and its density was noted down. Same thing was done with N14 type of normal DNA, and its density was noted down. And one more bacterium from that medium was taken from N15 and now shifted to grow in N14 medium. And after one step of replication, that means 20 minutes are gone, the DNA of the resultant daughter cells were taken out, and it was found by centrifugation that it was not as heavy as the N15 one. It was not as light as the N14 one, but it was somewhere in between. So this in between condition confirms that half of the DNA or one parental strand and the other one is a newly synthesized one. So the older strand is N15, and because the new medium given is N14, so the new strand synthesized would carry N14. This officially confirmed that DNA replication is semi-conservative. Similar experiment was done by Taylor and his colleagues in 1958, and they showed similar semi-conservative mode of replication with the help of Visia faba or faba beans. Root tip cells were taken, and the material they used for the experiment was radio-labeled thymidine. So this was the experiment. We learned in the last class regarding the machinery also and the mechanism and the enzymes involved for the DNA replication, that the process involves origin of replication, because replication cannot just start anywhere, it has to start at a particular place. That specific point is called as origin of replication, and in the case of prokaryotic machinery, origin of replication is just one, even in the case of viruses. But in the case of the eukaryotic, there are many origin of replication, so it's a difference between replication of prokaryotes and eukaryotes. Second thing we learned was the activation of deoxyribonucleotides with the help of the enzyme called as phosphorylase, which turns the MPs or monophosphates into triphosphates. And the second thing, the deoxyribonucleotides play two roles. First role is it acts like the raw material for DNA synthesis, and second thing is whenever this triphosphate liberates two phosphates, because it will join only as monophosphates to the newly synthesized DNA strand. So the loss of two phosphates will liberate enough energy to develop hydrogen bonds. So two functions are performed by deoxyribonucleotides as per the NCERT. Then we learned the role of the enzyme. First enzyme we understood was the helicase enzyme or unwindase, and topoisomerase or DNA gyrase enzyme. Then we understood the important role of the single strand stabilizing protein, which are supposed to stabilize the single strand, because we all know single strand DNA would be unstable and it will become reactive and may try to become again stabilized by joining with the complementary strand of DNA. So that can be prevented only with the help of these binding proteins. Then we understood that DNA won't open, the complete DNA will not open at one stretch, because that will be a very much energy consuming process, very expensive process according to the point of energy consumption. Therefore a small portion of DNA opens every time, and that gives the shape of a Y-shaped structure called as the replication fork. It's a request, please mention the 5', 3' polarities carefully, position of primer, and your arrows should be correct in both leading strand as well as the lagging strand, because these points are important while giving you marks in the board exams also. Every strand should be told which side is 3 and which side is 5'. Now the next is the enzyme. DNA polymerase enzyme cannot start the process, it can only continue the process. So to start the process there is requirement of a piece of RNA to be synthesized. That piece of RNA is called as primer, and the enzyme required for this is called as primase enzyme. Then the base pairing concept we understood. The ATP will again change into AMP, TTP will change into TMP, etc. with the help of the opposite enzyme now. So the previous enzyme was phosphorylase, now the enzyme is called as pyrophosphatase. And then elongation will continue with the help of the DNA polymerase enzyme, which has 3 types. And we understood the concept of leading and the lagging strand, and we understood that Okazaki fragments are also formed in this process. Okazaki fragments are formed in the lagging strand.